King Jesus, King Jesus. Now this is part 2 of my video, how mad scientists are creating new diseases to kill you. This is part 2. And I opened by introducing you to a prototype virus that is comprised of 3 parts. The capsule, which contains the nucleic acid or the DNA, and the attachment apparatus. So this is the prototype that we introduced. Now in this depiction of the bacteriophage that infects E. coli, you have 1 attachment apparatus per capsid. But that is not so in nature for every virus. Some viruses have many attachment apparatus on their surface capsid. There are many millions of different cells in nature, and for each cell there may be multiple viruses that attacks that cell. So there's a huge variation on the appearance of virus. These diagrams are magnified diagrams of what some of these viruses look like. You can see that some of them have few attachment apparatus on the capsid surface, and others have many attachment apparatus. Here's another diagrammatic depiction of the attachment apparatus on the surface of various viral configurations, so that you can compare them. But basically they operate on the same principles, and that's why the bacteriophage is used as a model for studying viruses, because they all conform to the same principles. They all work the same. It's the same mechanisms that they use. So that's why it's simple to work with bacteriophages, because it's more simple to understand. Now we also talked about the various phases of the virus infection life cycle on a cell. Here depicted as the bacteriophage infecting an E. coli cell. You have injection of the DNA, replication of the components of the new virus, assembly of the new virus, and the breakout of the new virus children, causing death of the cell. Okay, here we have a photomicrograph of a T4 bacteriophage virus infecting an E. coli cell. Now, you have to realize that this virus is specific for this E. coli cell, this type of cell, and this type of cell only. In other words, it won't infect other types of bacteria that may be in the gut. It won't infect staphylococcus cells. It won't infect streptococcus or enterococcus cells or H. pylori cells. It will only infect E. coli cells that are in the gut. And that is because of the attachment apparatus is designed only to attach to and infect E. coli cells. Even more important is that this virus cannot infect animal cells. It cannot infect human cells. It cannot infect any animal cells. Okay, this diagram shows you again how a virus infects a cell and ultimately destroys the cell by breaking new copies of itself out of the infected cell. Now, this is the case of a bacterium. Well, a human being is a collection of cells. And the Ebola virus, it doesn't attack all the cells in the body. It only attacks one type of cell. Those are the endothelial cells that line blood vessels. That's why you get bleeding. Once those cells are attacked, a person begins to bleed, and his death is not caused by the cell destruction per se, but by the bleeding that results from the destruction of the endothelial cells that line blood vessels. Okay, now this diagram gets into actually how the virus accomplishes reproducing itself inside an infected cell. This example is the papovirus that's infecting a human cell, and you can see that the virus attaches itself, infects the cell, and the DNA from the virus gets released inside the cell. Then the process of transcription and translation produces new viral DNA and new viral proteins that will serve as structural proteins for the capsule and enzymes to create new viral particles. Here the viral DNA is replicated and some viral proteins are made. And then in this step, you see that there are more viral proteins made to assemble the virus. Then the virus is assembled completely and is released to infect again. So that's the cycle of transcription and translation in the virus. And now I'm going to go into further detail about transcription and translation in the viral gene in the next video.